Biology chapter 19 theory video 3 we are going to discuss about reverse transcriptase. So now the using chromosomal DNA of a eukaryotic organism of the genes of a eukaryotic organism is going to be a problematic is going to cause problems because they are made up of sequence of bases called exons that code for sequence of amino acids, and in between the sequences of these bases introns, in between these sequences there would be introns, and they don't code for any production of amino acids. So when the RNA is formed by transcription, it has to be cut into fragments of the lengths corresponding to the exons, and it should be joined back again to produce mRNA. Then that would leave the nucleus. So then, to avoid using DNA with both exons and introns, mRNA of the cytoplasm is used to make templates to make DNA. So this was a possibility only when we found out about reverse transcriptase, which is an enzyme found in viruses that have RNA as their genetic material. So reverse transcriptase would use single-stranded mRNA as a template and free DNA nucleotides to form a single strand of DNA. So then this would be completed by using DNA polymerase to synthesize another polynucleotide to form the double strand, which is going to be the complementary DNA strand. So this would be abbreviated to say cDNA, that is the complementary DNA strand. So what happens is, first reverse transcriptase would be used to get the genetic material and produce a single strand of mRNA, and that is going to be the going to produce the free DNA nucleotide to form the single DNA strand. And using the DNA polymerase, the polynucleotide, the second strand is produced with the aid of the polymerase. The double-stranded complementary DNA strand would be produced. So this complete sequence would be known as cDNA, that is the complementary DNA. Now, to synthesize DNA, we don't actually, if we know the genetic code, we would be able to synthesize DNA from nucleotides without cutting it out of chromosomal DNA, or even by using reverse transcription. Even genes and even the complete genome. Genome is the entire set of genes that makes up a cell that is found in the nucleus, as well as the mitochondria or the chloroplast DNA. That is, if it is a plant. So the entire genome can be made by using we can synthesize DNA by using the genetic code. Then this sequence of the nucleotides we cannot do this all by hand. We have to use a complex machinery controlled by a computer. So what happens is, there would be a computer that would direct the synthesis of short fragments of DNA in a DNA synthesizing machine. You don't actually have to know details about DNA synthesizing machine, but if you know that there is such a machine, that is sufficient. So now what happens is, these short fragments would be produced, and then they would be joined up to produce a longer sequence of nucleotides, and they can be inserted into plasmids for the use of genetic engineering. So without cutting out from a chromosomal DNA. So this method will produce complete new genes. Now, for example, in the production of vaccines, and also to produce the genomes of synthetic bacteria, having millions of base pairs also have been produced by using this mechanism. Write down a note on this. Reverse transcriptase. When using chromosomal DNA, complications arise because in eukaryotic organisms, it is made up of sequence of bases called exons that code for amino acids, and in between these there would be introns, another set of sequence of bases which do not. When RNA is formed by transcription, these corresponding exons need to be taken out and joined together to form mRNA. To avoid this, mRNA from the cytoplasm is used as a template for making DNA. This is done with the aid of reverse transcriptase enzyme found in viruses that have RNA as their genetic material. This uses a single-stranded mRNA as a template and a free DNA nucleotide to form a single strand of DNA, and using DNA polymerase, another polynucleotide of complementary DNA is produced, which makes the double-stranded complementary DNA abbreviated to cDNA. Using the genetic code, DNA can be synthesized from their nucleotides without cutting it out of chromosomal DNA or by using reverse transcription. Genes and even the complete genome can be produced like this. A computer codes for a DNA synthesizer machine, and these produced genes can be inserted into plasmids for the use in genetic engineering. Right, so as you can see here, the reverse transcription from mRNA to form cDNA, DNTPs are deoxyribonucleotide triphosphates, that is adenosine triphosphate, thiamine, guanine, and cytosine. So as you can see here, there is the first one shows a single strand mRNA. Then add a DNA primer, free nucleotides, that is DNTPase, and incubate with reverse transcriptase. So it continues to move forward. Then synthesis of DNA strand by reverse transcriptase will take place. Then mRNA template will be removed by digesting it with RNase, and then add DNA polymerase and DNTPs, and synthesis of a new DNA strand would be produced. Then single-stranded sticky ends will be added for the insertion of cDNA into vector, that is the plasmid. So that will code, that is the new gene for coding of new proteins. I hope it made sense. If it does not, please let me know, then I will explain it further.